The overall goal of this experiment is to observe cell behaviors during lineage progression from neural stem cells to neurons or glial cells.
This method can help answering quick questions in the neurodevelopmental field, such as the role of symmetric and asymmetric cell divisions, cell cycle lengthening, and cell growth rates. The main advantage of this technique is that it allows the observation of cell lineage for long periods. This permits the observation of switch from neurogenesis to gliogenesis within a single lineage, and the direct comparison of sibling neuronal and glia progenitors.
Begin by removing the brains from 5 to 10 E14 embryos in a petri dish with cold dissection medium. Use forceps to carefully pull out the skin and skull and isolate the brains. While working under the stereo microscope, use dissection forceps to remove the meninges. Then, split the telencephalon in the middle to separate the hemispheres. To isolate the dorsolateral telencephalon, cut along the dorsomedial curve and paleo-subpaleo boundary. Transfer the dorsolateral telencephalon to a 2 ml tube with dissection medium on ice until all brains are dissected. After microdissection of the dorsolateral telencephalon, place the tissue in a 15 ml conical tube.